I'm Carl Ernst. I work at McGill University at the Douglas Hospital in Montreal. In my lab, I was contacted by a family who had a child with SETBP1 disorder. And, we're not always lucky enough to have the people come into the lab and meet with us and, provide us samples. But in this particular case, we were. So the family came in and we got to meet with them. And we got samples from the individual, and what really introduced us to it was being contacted, out of the blue by the family who introduced us to the disease, who introduced us to their son, and it was almost a half a day. We had a nice connection, we had a nice talk, and we really learned about the disease on that day. So I study neurodevelopmental disease. I'm a geneticist, stem cell biologist. But it was a nice fit between what the family's needs or what they wanted with respect to research, and then what we're able to do. So from that day, to be honest with you, was where we got into this. We're very interested in the gene, and we really liked the family who came and met with us. So we're working on the human side, so not in rodents. We're trying to understand what happens when SETBP1 is, one of the copies of the genes is affected. And what I mean by that is we make models from people with the disorder. Usually we'll take their P and we'll turn the P into stem cells, and we'll turn the stem cells into brain-like cells. And then when we have those, we're actually able to see the products of the gene, and we're able to try and understand what kind of pathways it's part of. So there's really a basic biology component of it. So how does SETBP1 affect brain development, for example? And by looking at, people who are either missing a copy or have mutations in one copy of the gene, we can start to understand what it might be doing. Now, we always sort of think of the basic biology of the gene in the context of where might this lead. The therapeutic aspects of these diseases are always of interest to us. And that is one angle that we're working on as we build these cell models from real people who have the disorder. SETBP1 disorder fits into a spectrum of diseases that are rare, so they don't affect that many people, and they're neurodevelopmental disorders, so they affect the brain and they affect children. So, from my standpoint, any disorder that fits into this category is very important from a societal standpoint. It's very important to the families who are affected. And especially in the rare disease category, there's not a lot of options, at least from research or from therapeutic development for these families. So we kind of fit into that. And SETBP1 disorder fits right into that important category that's really, it's understudied, it's underlooked at. I think lots of people think the gene's very important. We know it's very important. Different mutations in it have been found in a lot of different diseases. So why aren't we studying this gene more? Might be that sort of was the first thing that I thought of when I was, learning about the gene. It just has such a fundamental importance in the brain and in other parts of the body. So SETBP1 disorder sort of provides the opportunity to look into, a very important gene in human health. So I'm really happy to be looking at this, and it also feels like a new frontier of things that we're trying to figure out. There's going to be something sort of lurking behind the doors, discoveries that are just sitting there waiting to be made. So it's exciting from a scientific standpoint as well. We are making human brain models effectively of SETBP1 mutations. Because the technology to do this is so new, it's really an opportunity to really get at the basics, the fundamental aspect of what this gene might be doing in brain. So I think we know a lot about the gene from the blood studies, where this gene's very important in there, and again, linked to disorders there. But we are interested in what is the role of SETBP1 in human brain development. At the end of the day, that's what we want to see. So when does it get turned on? How does it drive differentiation of a cell? So cells start as sort of baby cells, and they have to become adults, but they have to be told how to become adults. And SETBP1 is clearly doing something there, but what exactly is it doing, and how is it? Does it play well with others? Is it driving other genes to be turned on, or is it part of really important molecular pathways? So there's just a whole world of questions to be answered. I think importantly, we know what questions to ask, because we know a lot about brain development, but we don't know where SETBP1 fits into brain development. So it's a very nice time to be studying it, because we have a lot of great tools, from genetic engineering to stem cell biology to just our basic knowledge about human brain development. So I think one of the most important things is just to know that there are people like me out there. So, you know, your child or sibling or niece has a rare disease, and people all have their own personal views of it and their own challenges, and every kid is different. But there are people out there in research hospitals and universities and probably elsewhere, working on the disease. And, I've become part of the community, SETBP1 community, in a scientific standpoint. And I think people are really committed, scientists are really committed to trying to figure this out. We interact with parents and families and hopefully their children as well. And it is personal for us too, even if we don't have a personal stake in it. We don't have affected children. It's a nice time to collaborate with families and to learn what they want, and then to try and bring scientific expertise, and they bring their personal views and needs on it. I'm excited about the novelty of using new technology to actually answer some of these questions, because I feel like even 10 years ago, we could not really answer a question about what is the role of SETBP1 in human brain development. But we're really in a place now to study a particular disease, a particular mutation, and in the right cell type. And the right cell type means it's coming from the individual themselves, because a disease is not just mutation X and this is you. Of course not. Everybody's their own individual, and they all have their own background genome, their own genetics, and all of that interacts with mutations themselves. We all have our own private genome. So we're now actually able to model the diversity of neurodevelopmental disease and SETBP1 disorder. One of the most promising things about it is just when there is a community of families where, something's very important to them. So, kids with the SETBP1 mutation, for example, just the fact that that community exists means it means studying the disorder becomes a lot easier for people like me, because, for example, suddenly you might have 10 families willing to donate cells for the research. Or if you're going to move to some clinical aspect of it, you can connect with that network who can then send it out on their private Facebook group or what have you. We need to be thinking big. We need to be connecting with families about what they want, and we need to be taking risks with the research. What's the maintenance function of this gene, and is there something we can do about it today? We know we can't do anything about what happened before with respect to a gene mutation, but is there something we can do now?